So, hi everybody, and welcome to the first official course on multi-scale brain circuit modeling using NetPyNE and NEURON. We hope this is the first of many, and we're very excited to have you all here. I wanted to start by saying thank you to everyone who registered to the course. It's been an amazing experience to see the interest that the course had. And the ones that actually got selected were amazing participants, and many of the ones that we couldn't get in were as well. So we thank you a lot for your interest. We know you're all really busy with your own research and your own work, and we thank you for being here. Also, a lot of thanks to all the tutors who have been working really hard to prepare the course. So over the last couple of weeks, we've been preparing all the slides, the material, the exercises, setting up everything, the Slack channel, and we're very excited to get started. And also I wanted to say, just mention the different tutors are from the universities that are listed here at the bottom. So we see SUNY Downstate from Yale, Nathan Klein Institute, and as well I included Metacell, which is the company that actually develops the software, one of the software tools that we're using, the GUI for NetPyNE, and they've done a lot of great work. And of course, thanks the funding agencies which provide the money to organize or to develop the tools and organize this kind of courses. So this course is actually part of the NIH grant that supports NetPyNE. As part of the grant, we said we would organize this kind of course every year. In theory, it was going to be in person, but then due to the pandemic and so on, we decided to move it to online. And I think I hope it works nicely. It seems that it's going to do. We also have several grants from other tutors, for example, for developing NEURON, for developing the reaction diffusion toolbox. Other grants just research-based, which use both NEURON and NetPyNE. We also have a grant from NSF, which is going to support part of the sort of supercomputers Google Cloud, which will host the NetPyNE GUI, which we are going to use in a couple of days. So thanks to all our funding agencies. Okay, so what are we going to see today? This is just the introduction lesson today, introduction lecture. So the first thing I'm going to explain is what this course is about. So I'll explain what we're going to see and why we think this is important. And the second part will be about how the course is organized, the schedule, Slack, the projects on the second week, and all the details. And finally, I will leave some time for questions and comments. Okay, so let's get started. So what is this course about? So I like to start my presentations with this quote from Obama, who said, "As humans, we have explored space, but we still haven't unlocked the mysteries of the brain. So when we actually do that, that knowledge will be transformative." And you know, for many of us here, I'm sure one of the motivations to study the brain is the fact that many people, according to CDC, around 1 out of 4 people suffer some kind of brain disorder, from schizophrenia, Parkinson's disease, epilepsy, Alzheimer's, some kind of motor paralysis. So the hope is that by studying the brain, we will be able to understand these disorders better, as well as develop some treatments potentially. So why is it so complicated to understand the brain? So we think that one of the reasons is that there are many different nested scales. So the brain involves the molecular level, for example, channels and synapses and molecules moving around. We have the cellular level with all its complexity in neurons, with all the dendrites, which include a lot of dendritic processing, nonlinearities. Combinations of these cells then form circuits, local circuits, and when you get with very specific arrangements, actually connectivity is very specific for different parts of the brain. And then you can group many of these circuits into different systems or regions, for example, the visual cortex, the motor cortex. And overall, the whole orchestration of the brain together at some point gives rise to behavior, which can also, in a way, be considered part of the brain, as it is the main function of the brain is to generate behavior. So the complicated thing is that not only that there are many nested levels, but also that they interact with each other, and they interact with each other bidirectionally. So the complicated machinery inside the cell, all the molecules moving around, ionic channels, synapses, they in turn generate the action potentials, which is, we think, one of the main ways of communicating in the brain, as well as plasticity and other things. Groups of these neurons with very complicated inner structures then give rise to these local circuits, what we call the mesoscale. And that again has very complicated dynamics. The circuits are formed in the very specific structures, like the 6 layers in cortex, for example, with very specific connectivities. You can have excitatory neurons, inhibitory neurons, different responses. So it's all very complicated as well at that level. And then you form these regions. So the brain has many regions. For example, the cortex, if you spread it out, is sort of the size of a pizza, they say, with around 2 millimeter depth. And it's divided into many different regions, prefrontal cortex, visual cortex, motor cortex. But it seems they all interact with each other. And there is also like a very complex organization at the whole brain level with many of these regions interacting. And what's interesting is that, for example, this activity at the whole brain, they can generate these synchronized oscillations that travel the whole brain. That will affect in turn the activity at this middle scale, the mesoscale, these local circuits. So the activity in this intermediate level depends on the whole brain activity. And in turn, that activity in the local circuit will affect the single neuron dynamics, the internal dynamics, what happens inside, the plasticity, and so on. So all these different scales are all the time interacting with each other, and that is basically what generates the brain function and dynamics that we see and we use in everyday life. So how can we figure this very complicated object? So we're lucky that nowadays we have an incredible amount of experimental data, and it keeps coming out. We keep developing new techniques, new methods, both for recording the activity of the brain, as well as for recording the sort of anatomy, the shape of neurons, their connectivity, their pathways. So there is an immense amount of data. However, making sense of that data is not always easy, as there are many different labs, many different experiments going on, which are isolated. And that is where we believe computational simulations come in. So the idea is that we can take all this experimental data, combine it together, and try to make sense of what it means. So simulations allow you to have sort of a theoretical framework where you integrate all these different pieces of experiments and try to make sense of them. The basic idea is that we try first to model mathematically the different experimental data sets that we have. For example, the behavior of a single neuron, or the behavior of a circuit composed of multiple neurons. And then we have to validate our model so that it actually is useful. We have to make sure that what we have modeled actually reproduces the behavior that we're seeing in the brain, the function, the dynamics. And then we can use the simulation to try to understand some aspects of that piece of the brain that we didn't know before, some mechanisms, these different scales, how they interact with each other. We can also test out hypotheses. Well, we believe this works in this way, so we can go to the model and very systematically try different things in the model and check that it's true. And potentially, of course, we can simulate disease and try to develop treatments for those diseases. So why use the neuron simulator? So as you know, there are several simulators out there. However, the neuron simulator is established as the most appropriate for this multi-scale modeling. So modeling that involves all these different scales that we were talking about. So it includes, at the lowest level, modeling of the reaction diffusion that occurs at the molecular level. We can model in a lot of detail the different ionic channels, synapses, the morphologies of neurons, and we can also combine many of these neurons to form more complex circuits and very large circuits. So the Blue Brain project has, for example, a model in neuron of the whole mouse cortex, which is millions of neurons. So why the Neuron Simulator? Also because it's one of the most widely used. So if you go to ModelDB, this database, you will see it contains mostly models in Neuron. It is the most cited simulation tool as well. There's new papers coming out every year that use Neuron. There's currently over 2,000 papers published that have used Neuron. There's a complete list in the website. And there are many existing models. As I mentioned, in ModelDB, you can find a list of all the models in Neuron, and you can borrow pieces and components from them. So you can borrow cells, you can borrow ionic channels, and so it's a great tool to use because we have a lot of resources available. As I mentioned, it's also used by some of the main modeling projects in the world. So the Blue Brain project, for example, released this model of the rat cortex S1 around 6 years ago. It's since been extended. The Human Brain project is a $1 billion project in Europe, one of the largest in history. Also develops, for example, this hippocampus CA1 model in Neuron. And the Allen Brain Institute has also developed recently this model, very detailed model of the mouse cortex, primary visual cortex, B1. So what is NetPyNE and why do we need it? So all of these models that I was showing, they all have these big companies or research institutions. They have their own tools on top of Neuron that help you build these models. In some cases, these are sort of private tools that are not shared. In other cases, they are shared, but sometimes they are quite complicated to use. So this infrastructure on top of Neuron is usually some kind of Python package that helps you build these very complicated models. And that is essentially what NetPyNE is. So it's a Python package that helps you develop, run parallel simulations of these models, optimize the parameters of the models, which we'll talk about more later, and also analyze the output of these models and the structure and so on, all built on top of the Neuron simulator, which is used as the backend simulator. And keeping on with this discussion of why we need NetPyNE, sorry. The idea why it started is that we were trying to build one of these very complicated models, in particular one of motor cortex, and we were trying to combine data at many different scales, so it turned out to be very complicated and very complicated to keep in a simple way. So we started developing this tool in our own lab to help us combine this different information of different cells, the morphology of different cells, connectivity at different scales, and so on. And then gradually it became a more widely used tool, and we started sharing with other labs. So the other reason is that we wanted to make it as simple as possible to define these models. And we wanted to keep a separation between sort of the biological parameters from the underlying low-level code in Python. So when you code in Neuron, you have to use some code to create the different structures. And in the middle of that code is usually where you find all the biological parameters of what you're creating. So in NetPyNE, we wanted to keep those as separate as possible. And so we created this declarative language where the idea is that you define only the biological parameters of what you want to construct. So for example, a population with 200 cells of this specific type with these channels, and that connects with a probability of connection of 20%. And then NetPyNE takes care of converting that declarative language, that specifications, into a neuron model. And you don't have to worry about all the more low-level implementation and code. So the idea is that by using a declarative language, you also make it easier to share models in the future, because they will all be implemented in the same standard language. It is easier to interpret and read other models, to share them, and to try to reproduce them yourself, to modify them. For example, if you go right now to this database of models in Neuron, they're all implemented in very different ways, and sometimes it can be quite hard to figure out what the user was trying to do and what the actual parameters of the model are. And finally, we also wanted to make it easier to run parallel models. So of course, this is possible in Neuron, but it requires some knowledge, and you need to know how to divide the neurons among different nodes. You then need to collect the output data from each of the nodes and combine it together. And as well, we wanted to facilitate running these parameter explorations or optimizations that are very common when you build models to try to fit the model to the actual data. And we wanted to make that easier as well. So this is an overview of the NetPyNE package. We will go more in detail later on in the course, but just the basic idea is that you have a first step where you define the model, what we call the high-level specifications. You then create a NEURON model based on those specifications that we call the network instantiation, and you can then run parallel simulations. Parallel simulations where NetPyNE takes care of, again, distributing the cells, gathering all the data, and afterwards you can use a number of built-in analysis functions in NetPyNE that allow you to do common things in neuroscience, like plotting connectivity matrices, raster plots, spiking statistics, LFTs, saving to different formats, exporting to other simulators, all within the NetPyNE package. And as well, here at the top, we have this batch simulation module, which is what I mentioned, to facilitate exploring the parameters of the model in an automated way. So just to clarify the relation between NetPyNE and NEURON, here NEURON is always at the bottom. It's the sort of backend simulator tool that we're using. This is some example code in NEURON where you would use this code to create a synaptic connection. Allow someone in, okay. And on top of NEURON, we add this new layer, which is NetPyNE, which has this declarative language, where, for example, you would specify a connectivity rule. You want to connect the sensory population to the motor population with a probability of 50%, a strength of 0.1, and so on. And then NetPyNE would take care of generating all the code necessary to create these connections. And finally, on top of this, we have the NetPyNE GUI, the graphical interface, which basically allows you to do everything you can do programmatically using this kind of code from a graphical interface. So we will see this more on this later on, but basically this standardized declarative language in NetPyNE allows you to define all the components of the model, the populations, the cell properties, synapses, connectivity. And it also includes support for this reaction diffusion module in NEURON that we will see more about on Tuesday, which essentially allows you to model in more detail all the internal processes and external processes of the neuron at the molecular level. And that is interesting because then you can relate these molecular processes to the network level. So for example, you can see how second messenger cascades or buffering affects the firing of the network or plasticity and these kind of effects. For parameter exploration, we also see examples on the last day, and the idea is that you can run these, set up these explorations in a very easy way and run them in different setups, for example, on supercomputers or on your local machine if you have a big machine, and everything is sort of automated. So, for example, we have used this in this NSF grant that I mentioned before to run simulations on 100,000 simultaneous cores on Google Cloud to explore one of the models we were building, which was quite a detailed model that required a lot of cores to run, and we needed to explore the parameters to make sure it was similar to the experimental data. And so these supercomputers allowed us to run on these 100,000 cores simultaneously to actually do this very fast. And this was all using NetPyNE and the NetPyNE parameter exploration tools. We also have what we call evolutionary algorithms, which allow you to, again, fit the model to experimental data in an automated way. And as I mentioned, there are many built-in analysis functions, so you can plot from these different connectivity views of the network, voltage traces, raster plot with the spike times, and statistics, for example. You can also use NetPyNE to record local field potential, so you can actually place an simulated electrode in any region 3D space of the model. So you can put, for example, electrodes at different depths in the model and record the LFP signal in a pretty accurate way, pretty realistic way, so that you can compare to experimental data. So LFPs are obviously very common in experimental data sets. And you can also now calculate current source densities, which is sort of another analysis based on LFP data, and also very common in experiments. Here we have a view, a screenshot of the graphical interface that I mentioned, and we're going to see on Wednesday. So this allows you again to do the model building, simulation, and analysis from a graphical interface. And it's quite useful to start with because it provides you an overview of all the features that NetPyNE provides. And it's easier to interact initially and to learn about the tool. You can also then export, for example, from this graphical interface, you can export to a model, to a script, to the programmatic code, so to speak, and you can continue your model programmatically once you have built it initially using the GUI, for example. Another important feature of NetPyNE is that its standardized language allows you to export to other common standard languages, a standard format, sorry, like Sonata, which is the one developed by the Allen Brain and the Human and the Blue Brain project, as well as NeuroML, developed by UCL. So this, in theory, allows you to share your models with other tools. So here you see, for example, NetPyNE, and you could export the model and read it in Brian or BMTK, INN, some of the other tools available using this common format. This is an example of the same network model read from a Sonata description in NetPyNE and some other simulators, and it produced exactly the same output. NetPyNE has a lot of documentation and tutorials. You can go to the website, netpyne.org, and see more about it. We also have Q&A, question and answer forums with lots of questions from different users. So if you have issues, you can also check here initially. We have the eLife publication with more details on the tool. And importantly, we are very happy that there is a sort of growing community of users in NetPyNE. So we have tracked about over 40 labs have used the tool, and there are over 80 models that have been developed or are being developed. There's a full list here in netpyne.org/models. So it's very nice to see that there are models of many different parts of the brain, different cortical regions, cerebellum, even thalamus, inner structures like claustrum, as well as different disease and disorders like ischemia, epilepsy, and even different treatments. So we have some people at Duke University, for example, who are modeling the effect of TMS and TDCS. And by the way, we will see on the second week of the course, we will have 10 invited speakers who are users of NetPyNE and have developed models using our tool, and they will provide a nice description of what they're doing. So that I think that will be very interesting for all of us, especially as well as for me, because I haven't seen the details of what they're doing. So I'm very excited about that too. And to finish off this section, I wanted to give an example, a specific example of one of the models that we've been developing for a while, and that illustrates this idea of integrating experimental data and how we can use modeling to gain some insights into brain function and brain structure and dynamics. So this is a model of motor cortex circuits. And so here I'm just showing some example experimental data where the different cell types in the 6-layer motor cortex circuit are divided by the target that they project to. So different cells in different layers, they project, send axons to different parts of the brain. And the main 3 categories are these IT cells, which are basically, they're called intratelencephalic. They project inside the telencephalon, inside cortex mostly. ET or PTs, which are pyramidal tract projecting, and they project out of the telencephalon down to the spinal cord. They're sort of the output of the system, and then the corticothalamic, which project mostly to thalamus. So you can see a nice representation also overlaid over the mouse brain of these 2 of these different cell types. So the PT neurons start here in M1 in cortex and go down to the spinal cord, the pons actually, and these IT neurons which project within the cortex, and they're actually projecting in this case to the other hemisphere. So what we did is combine a lot of this experimental data from different labs and different scales into a single model. So we combined data from over 40 different papers, as well as our direct collaborator, Gordon Shepherd, which provided a lot of the data. And so we constructed a model, a detailed model of a motor cortex sort of cortical column. Here it's illustrated in the mouse brain, sort of the representation of what we're modeling. It includes around 10,000 neurons, 30 million synapses, and you can even see here in the diagram the different cell types. There are these IT cells that I mentioned are in red, the PTs in deeper layers in blue, and CTs corticothalamic in green at the bottom. So this is actually just showing 5% of the neurons, just for illustration. But the circuit itself is much more deeply packed. So this illustrates a bit of the detail that we put into the model. So we included, for example, the different layers, the different depth of the layers taken from experimental data, the densities of neurons in each layer. They're also very specific to mouse motor cortex. We have the different cell types that you find in the different layers. We have the three excitatory cell types, as well as 2 different interneurons, inhibitory neurons. So for each of these cell types, we actually did a very in-depth process to match them to experimental data. So for example, this is this layer 5B corticospinal neuron. So we see here the experimental data, the morphology that we based our model on, and the actual simulation, the model of this morphology. Also for the actual neurophysiological response, we see that it's very similar. Here is experiment versus simulation. And even what we call the subthreshold activity, when the neuron is not spiking, we also matched it quite in detail. And to do this, we required running these optimization algorithms for several months on supercomputers, where we were basically modifying the different ionic channel densities of the neuron model within biological constraints to match the neurophysiological response of the neuron. So we did this for all the different cell types involved, and then we combined them into a circuit also based on a lot of experimental data. So connectivity data came from first long-range inputs, so different parts of the brain that actually provide input to this motor cortex circuit, and different parts of the brain project to different parts of the circuit. So for example, sensory thalamus projects mostly to the upper layers, layer 2, 3, whereas M2, the secondary motor cortex, projects more to the deep layers, as well as to specific cell types. We also include a lot of information on the local microcircuitry. So within the 10,000 neurons that we have, how do they connect to each other? And we had a lot of detailed data from Gordon Shepherd on this. And we even had data on the distribution of the synapses. So maybe a neuron can have thousands of synapses coming in from different neurons. So how do they distribute along the dendrite of each neuron? And this is specific for each projection. So for example, neurons in the layer 2, 3 project to the upper part of the dendrites of these layer 5 PT neurons. So we put that kind of rule into the model, and we combine all these different rules for the different projections into a single model using NetPyNE. So I'll show briefly some of the results we got from this model. So this is showing a raster plot. Shows the spike times of the 10,000 neurons along a couple of seconds. And what we did is try to match the statistics of the different populations. So for example, here you have the IT cells in layer 2, 3. We tried to match them to, oh, sorry, my headphones died. So we tried to match them to the different experimental data sets that we had. So for example, for layer 2, we had this data from a paper from some guys in the UK. And again, so we did that for each of the populations. And so we see that they're statistically similar to the experimental data that we have, based on both the cell type and the layer of the neurons. We also reproduce some of these LFP oscillations. So we have,
for example, oscillations in the delta and the gamma frequency. And these oscillations arise spontaneously, so we didn't really enforce them in the model. But due to the connectivity and the realistic biophysics of the neurons, we got this kind of very similar oscillations to some of the ones you see in motor cortex. And even this idea of phase amplitude coupling, where some of the amplitude of the fast oscillation depends on the phase of the slow oscillation. So some of these mechanisms that are very specific to brain and are related to neural coding, they showed up in the model as well. We've also been able to relate this to different behaviors. So the dynamics in the mouse motor cortex when the mouse is moving or when the mouse is quiet, stationary, are different. And so we have matched that to the different different states in the network model and compared it to experimental data. So what was good about the model is that now that we have reproduced some of these oscillations, we can look into what are the mechanisms behind the generation of these oscillations. And so, for example, the model revealed that there were different populations oscillating in antiphase, and that seems one of the causes why these oscillations are happening. So we can then, for example, try this as a hypothesis and record experimentally from these different populations and see if this is actually true. So the model provides some insights and allows to generate predictions. As I mentioned, we also looked into different states or different behaviors of the mouse, and we tried to reproduce a specific paper which showed that with noradrenaline modulation, the mouse behavior was sort of correct, precise, whereas without this noradrenaline modulation, which comes from a region of the brain called the locus coeruleus, the mouse paws were not placed correctly in the wheel. So we simulated the different states, and then using the model, we could come up with a prediction of why this noradrenaline modulation was generating different behaviors. In particular, it seems like noradrenaline blocks the HCN channels in some neurons, in particular in the PT neurons. This increases the firing of these neurons, which leads to precise movement. So here we see the model simulating the different states. Here at the beginning is the quiet state, and here is the movement state. So we change some of the variables of the model that we know happen. We know there are differences in these two different states, and we simulate in the model. And then we compare also with experimental data from the mouse in these two different states. And then we simulate again this sort of not healthy state in the mouse where we have blocked noradrenaline, we have blocked the inputs from this locus coeruleus region, and we simulate also in the model, and we compare to recordings from the mouse. So this basically is linking this molecular level where we are looking at the noradrenaline effect on the HCN channels on a specific cell type of the circuit and how that affects the dynamics of the whole motor cortex circuit. And we know that these different dynamics are linked to behavior. So we're basically here trying to relate all the different scales that we were talking about, from molecular, the cells, the circuit activity, up to some measure of behavior. And again, what was interesting about the model is that it also provides us with more in-depth understanding of what's going on. So the experimental data set here in the top left, we had some data from neurons in layer 5B, but with the model, we could decompose those different neurons into different cell types that you find in layer 5B. So there are the IT cells, the PT cells, and even the upper and lower part of the layer behave differently. So basically, the model was providing some insights and some predictions of what was going on within this layer 5B activity that was related to the behavior distresses. So I hope this gives you some insight, some understanding of some of the things you can do with these kind of models. And now we're going to move on to the second section. So we're going to see how these how the course is organized and look at some of the logistics. Okay, so I wanted to start just by showing some of the statistics of the people who applied. So we had 87 applicants from 21 different countries, 6 continents. So we were really excited about this. We forgot to include in the form sort of a gender field. So just for statistical purposes and for our grant funding, we sort of estimated the percentage of male versus female. We know that this doesn't cover all the different genders, and we apologize if some of you don't feel identified with any of these. But this is just sort of an aggregate measure for statistical purposes. And we are quite happy to see that, well, at least a third of the applicants were female, which is relatively uncommon in this field. We also had a very large range of sort of professional status, so from undergrad students to quite senior faculty. So we decided to keep only 20 applicants, as stated on the website, because we wanted to provide, focused attention and personalized attention as much as possible. And so we based the selection exclusively on the form that you provided, so on the background experience that you had in neuroscience, on your Python programming experience, and in the proposal of your project. We must say that we were very, very impressed with the quality of the applicants. Their experience, the Python background, the proposals of the projects were super interesting. So it was really tough to make the decision. We basically gave scores to the different applicants, and then we just added the scores and selected the top 20 participants. So we were quite happy with the result because we didn't really force any kind of diversity or gender balance, but the result was that we got still 20 participants from 8 different countries, 5 continents, and very interestingly, 60% of the selected were female, out of only 1/3 of applicants. And again, we kept some of the diversity in terms of status, so from undergrads up to faculty. So these 20 participants are the ones that on Slack will have the S of student. And we also have, including the course, some new members of the different labs that are organizing the course. New members and some collaborators that we were hoping to train on the methods and tools that we use. So we decided that this was a great opportunity for training them. And so we have included them here in the course. Those are 17 additional participants that you will see with the N on Slack. And these, again, range from high school students, undergrads, PhD students, postdocs, some software developers, and some faculty. I just included here because it's interesting. Most of them come from the US, but we do have one from Argentina, from Sierra Leone, from Australia, and from Chile. So some diversity there as well. And I wanted to also introduce and show pictures of all the 10 tutors for the course who will be teaching and helping you in the second week. So these are all experts in building these kind of models, either using NEURON and or NetPyNE. They will be available on Slack, and they're happy to help. Here they are ordered alphabetically. And I hope we have time during the course, maybe at the end today or whenever we have some spaces left so that they can briefly introduce themselves. But in any case, we do have all the introductions on the Slack introduction channel. So the schedule, so we have the 5 first days of this week. They are quite intensive. So day 1, so today and tomorrow will be mostly NEURON. Today is mostly single cell NEURON with some theory before that will be provided by Bill Litton. Tomorrow we'll have some description of the reaction diffusion module in NEURON. as well as some implementation of networks and synapses in NEURON as well. So we cover both single-cell reaction diffusion and networks in NEURON during Monday and Tuesday. And then Wednesday to Friday will be NetPyNE. So we will start on Wednesday looking at the GUI, because as I mentioned, that's quite an intuitive way to get introduced to the tool. We will then do some tutorials, more programmatic tutorials in NetPyNE. We'll look at some specific features like LFPs, for example, and also the more internal data structures of NetPyNE. So how is it organized internally? What happens when you import a cell? And Joe will also provide some overview of the different plotting and analysis functions that we have. And the last day, we will go over a bit the idea of building these more detailed models based on experimental data. How do you go about that? How do you gather the data? How do you run these parameter optimizations to match the model to the data? And also, Craig Kelly will provide an intro on how to use the RxD module in NetPyNE so that you can combine these molecular levels up to the network level. So the last hour of each day will be mostly dedicated to hands-on exercises. This will be like guided, so you will get some instructions, and you have to do the coding yourself, and we will help you and give the solutions. We're going to use Google Colab for most of these. So we have like a session today that introduces Google Colab and how to use it. And during many of these tutorials as well, and during these lectures, you will have some kind of demo that you can follow along. So many of these will also be in Google Colab, so you might get some theory at the beginning of the class, and then at the end some demonstration that you can try yourself on your laptop. And the second week will be mostly dedicated to the projects, to building the projects in groups. I will talk about a bit more about that in a minute. And those will be interleaved with speakers. So we'll have 2 speakers every day. Again, people who have been developing models in NetPyNE for some time, and they will show the current status of the model. And that will go from Monday to Friday again. And then on Tuesday, June 1st, will be the last day of the course. And we will have the presentations of the different projects by the different student groups. So a bit more on the lectures and tutorials. We will use Google Meet, as you have seen. I will share the link on Slack. The idea is to use the same link for the whole week. So this is the same link that I shared today. You can just use that for the whole week. If there is enough bandwidth and there's no issues, please turn on the camera, because it's nice to get some visual feedback. Yes, I mentioned there will have hands-on exercises that you can follow along. The last hour will be hands-on exercises. We will have these breakout rooms. So while you're doing exercises or during the lectures, if you have any issues, we can separate you in a small room with one of the tutors to help you out. So just let us know either on Slack or here in the chat. And please feel free to ask questions anytime. We will usually leave some time specifically for this at the end of the lecture, but you can, of course, anytime post questions on the chat, Slack, or just speak up. And as I mentioned, this will be recorded and made publicly available, hopefully the same day or the next day for those who cannot attend in person. Okay, so a bit more on the modeling projects. This will be mostly done during the second week. Of course, you're free to start before if you find the time, but we know the first week is quite intensive. So we were going to propose that, if possible, you do the projects in pairs. This is both because I think it will be easier for you to discuss among you, to answer questions and look for what you have to do in pairs is usually easier. And as well for us, to help you, we only have 10 tutors, so it will minimize the amount of work required and provide more tutor time for each group. So the modeling projects are pretty free. Oh, I think Bill has raised the hand. Bill.
I just wanted to check if raise hand worked. Sorry. But that's good that it alerts you, because that seems like a good way to have people interrupt and ask questions, which would make it whole thing more lively.
Great. So it works. So the projects, they're pretty free, so you can do. We're going to be very flexible. So the idea is you can, for example, build a toy model of some brain region, of some phenomena, of some disease that you're interested in. You can also just start building the scaffold or the basics of a larger model. So, of course, these models that I've been talking about take a long time to build. We've been building one of the auditory cortex for over 1.5 years now. So, of course, in a week, you're not going to have time to do much. But I think it's also completely fine to just start building one of these complicated large models. So in the project presentation, you don't necessarily have to show some fantastic results, but you can just tell us about how you're building from the bottom up this detailed model, how you've been looking for information, how you're going to go about it. So this is also an option. You could also get one of the existing NetPyNE models, which we mentioned there are like over 80 available, and do some modification to it, extend it in some way, improve it. Or you could also take another existing model, either implement it in NEURON from ModelDB, or in another simulator, like in Nest, for example, and convert it to NetPyNE. So this is something quite common that has happened in other courses that we've done, for example, in Brazil. And it's also very useful to learn and to have the model in NetPyNE, because other people can then use it more easily. So each group will be assigned one main tutor. We will try to make those decisions based on your interests and some suggestions from yourself and from the tutors. And the idea is that these tutors will sort of keep track of your project, and ideally, during the second week, meet with you at least once a day, maybe like a short, 20-minute, half-an-hour meeting, just to see how you're doing, if you need any help, and so on. But of course, we are encouraging you to talk to as many tutors as you can, to other students, just interact with everyone you can on Slack, and ask questions, and have meetings with the different tutors, and so on. So we're be completely open about interacting with everyone you can. So finally, the presentations will be on Tuesday from 10 a.m. to 6 p.m. We decided around 15-minute presentations with 5 minutes for questions afterwards. Questions will come mostly from the tutors, but we'll also open it to the other students. The order of presentations will be selected randomly. This was a suggestion from Joe, who apparently this happens in the Japan course, and it forces students to actually pay attention to the other project presentations. And we decided that all tutors and all students will give a score to each of the project presentations. This will be done anonymously via Google Forms. And then we will give 2 prizes, so based on the highest score by the tutors and the highest score from the students. So these 2 groups will receive a fantastic prize, which is either registration to the Computational Neuroscience meeting, the CNS meeting, which this year will happen online as well. So in case you didn't have enough with this course, we will force you to go to another computational meeting. And if you are not interested in this, or you already registered, or whatever reason, we can also give you some book, which will be selected by one of the invited speakers. And for all of you that complete the course, which basically means just giving a project presentation, you will get also a certificate of the course completion. And finally, have fun. So unfortunately, this course is not in person, so you cannot do things like we've done in other courses, like the one in Brazil here, where you interactively go to the board and answer questions, or go for some drinks. But this is some pictures from the CNS 2020, and it can also be very fun to do it online. You can see all these very happy people here. Specifically, you can just focus on Bill's face, which is one of the tutors here, which is really excited. No, but kidding aside, actually they can kind of be fun, and we really encourage you to talk as much as possible with other people and learn from others. Like the The introductions that we've seen in the Slack are really, really impressive. So I really encourage you to talk to other people and learn from the tutors and from other students. So any questions, comments? I hear something.
When's the best time to start just chatting? Is there a forum to start chatting with you guys, with the tutors and other students, sort of informally, not recorded, without a presentation, where we can start having some chats about what we want to do?
So we thought that we would have like official meetings with the tutors on the first day of the second week, once we have assigned tutors to each group. But that said, if you've seen some in the introduction channel, for example, someone that you think is has similar interest to you, and you want to chat to them, just use Slack, chat to them in private, and if you want, try to organize like a video chat during this week, sometime that you both have time. So yes, feel free to be very natural about this. Just contact anyone you want and chat to them. We're all here and excited to meet you and to talk to you. So please go ahead. I just realized I had a slide on the Slack channel, but it must have disappeared. Okay, but I think
maybe you could give an introduction to Slack and also to Google Meet. I actually just was wondering who asked the question. How do I see who asked that question?
Oh, I have no idea. I just saw him here in the screen, but.
Oh, you did? Okay. I mean, because I turned on the chat. Oh, forget it. Okay.
Yeah, I have like a list of all like a grid view with all the faces. And so you see whoever puts the hand up, you see like a little hand on the bottom corner, and then you see them speaking. So Slack, yeah, I lost my slide, I don't know why, but basically we have several channels. Let me see if I can get it here.
Maybe just put Slack up and show them typing into Slack.
Yes. Let's see. Share.
So one other comment, by the way, sorry, hi. If you put on closed captioning, it will tell you who's speaking.
I just did that, yeah, thanks.
All right, now somebody has to speak so I can see the captions. Oh, there they are.
I think you're seeing now the Slack. Okay, so just a quick overview. Here on the left-hand side at the top, you have a list of the channels. And we already described this by email, but essentially we have the general channel where we will make announcements for the course, like the links to different things, or general announcements that everybody has to see. The introductions channels, which I think most of you have already posted, a nice brief paragraph explaining your background, experience, and interests. So if you're interested in knowing something about any of the participants, you can just go here and you can search. Here at the top, you can search for a name. And it should be a name. No. I have to type the whole thing. It will show you the introduction from this particular person. The Only Students channel, this is just for you guys to talk between you. I'm gonna exit the channel as soon as I add everyone here. I think there's 2 people missing, which we're gonna find out.
Just for making fun of Salvador, I think.
Yes,
right?
exactly. You can make fun of me and all the tutors here, and we promise not to look. And so we have 2 people missing. As soon as I find who they are and add them, I will exit the channel. The Only Tutors is private for us. And then we have the water cooler, where you can post anything you want, from interesting things on neuroscience or anything you find funny or interesting that you want to share. And the Ask Tutors channel, which I think will be the most active, where you can post any questions you have about the course, about neuroscience, about the tools, and we will make sure we answer them. So we have, Slack has this thing called threads. So if you go to one of the messages, you see these pop-up icons in the top right. For example, you can add a reaction, which I think you've already learned about. But you can also reply to the thread. So if you click here, you will see a sort of a private set of messages just for this question, for this message. And you can keep a conversation about this topic on a separate level here. And then you have this direct messages section where you can talk directly to any of the participants in the course. If they don't show up in your list, you can just click here on the plus sign and type the name, and you can talk to them directly. Unfortunately, this is not a paid version of Slack, so you cannot use the video call feature. So to arrange video calls with other participants, you can either use your own favorite tool, if you have Zoom or whatever, or if you don't have access to any or you prefer, you can just let us know, and we will create a Google Meet specifically for you that you can use.
Salvador, it's worth mentioning in the direct channels, I think this is true for even the free version, you can bring in 2 or 3 or 2 is standard, and then you can bring in 3 or 4 people and maybe show how to do that.
Yes.
Because that would be most useful for the projects. You'll bring in your partner and the tutor.
Yes, exactly. So if you click on this plus sign that I mentioned before, here you can create a list of users like Bill said. So you can say, for example, I want to talk to Robert. Click here, as well as to Bill and to Don. You click everyone you want to talk to, and then you just start talking. And you have this sort of private channel with these people here that will stay here on the left-hand side, and you can use it to coordinate your projects or some other conversations you want to have.
But a lot of times it's good to have the conversations in public. So I think we can make additional channels for sub-projects and groups, so we can sort of put people into those.
Yeah. We can definitely do that. Once the projects are sort of assigned and the groups are assigned, we could create channels for each project if that is useful. So.
Oh, and one other thing, yeah, to make sure somebody, if you want to get somebody's attention to a message, use an @ symbol and then type the first couple letters of their name and click that, and then it'll draw their attention to that message. Otherwise, messages have a tendency to get missed.
Yes. So for example, here, if I want to talk to Joe, I would click the @ symbol. Joe, hey, help me.
And now I just got a bleep on mine saying, Joe, you got a message.
I guess I wanted to encourage Joe that they feel free to speak frequently and freely on the Slack. I feel that people are sometimes intimidated by having their really silly sounding questions, which usually turn out not to be that silly anyway, but they feel they're very silly questions asked in any public forum. I guess one use for a little bit of privacy to be able to feel silly without losing face.
Yes, usually the silly questions are the most useful because everybody wants to ask them and they don't dare. Yeah, so any other questions, comments from students, tutors, anyone?
There's still some raised hands. I guess either you can speak or unraise your hand.
Yeah, feel free to speak.
Robert, your hand is raised, or am I just not updating frequently enough?
Maybe he's stretching.
Yeah, I was stretching.
Okay, so I was sorry, I was expecting to have the word. Well, hello everyone. I'm Javier. I'm just going to write my presentation, my proper presentation on Slack. But I want to. Okay, so first, thank you all for having accepted me here, and I wanted to have a comment on the last day presentations, because even though this particular course I'm in line with the time zone, I don't know if other people will have troubles with this time zone and the random assignation, which I think it's a great idea. I don't know if it's going to make them harder to participate or not? So maybe I wanted to propose that you got this into consideration. That's it.
Yes, that's a very good point, Javier. Thanks. We
Thank
will
you.
think about the different time zones and limit the slots for those who are in a weird time zone.
I don't think we should call other time zones weird. So
They are weird.
that's probably denigrate.
Non-US
I
are
think they're very good time zones.
Yeah, I'm actually the second week I'm gonna be in a weird time zone myself because I'm gonna go work from Spain. So, okay, other questions?
So Salvador, you did assign a lot of time for each of the talks, including your own. We're going to take these long breaks, I guess. And most talks will be an hour, I would imagine.
Yeah, so I left some time at the end, like if there are questions or you have exercise that people are doing so they can finish off. But if not, you can go to break before time, yeah. We only have like 10-minute coffee breaks, so maybe if you have extra free time at the end, we can have a bit more of a break.
And happy hour break, don't forget.
Yes. Yes, Sydney?
I was just going to ask a sort of more technical thing, whether you were talking about the LFP recordings. I wondered whether anyone has created or is thinking about other common functional ways of recording activity in vivo, so like calcium imaging. Have you got outputs that can simulate that sort of data?
Stay tuned for tomorrow's episode with Robert.
Okay.
So, yes.
Yes, calcium imaging, it's not sort of built into NetPyNE, but it's possible to do, of course. And we do have also ways to reproduce EEG signals, simulating current dipoles. And we're also working on simulating fMRI-like signals, like the Bolt signal, which eventually will be built into NetPyNE as well.
Yeah, eventually these things will be in NetPyNE. And for those who are really programmers at heart, that could be a project, is to just get something that's currently not immediately accessible in NetPyNE. It's still accessible via NEURON code, but to get it into a very easy to use format in NetPyNE would be a nice project. It would help us.
Love that idea.
Any other questions, comments? So if we had spare time, I thought it might be nice if the tutors introduce themselves very quickly, like 1 minute each or something. So I'll just quickly start. So my name is Salvador. I'm originally from Spain. I did my undergrad and master's in Spain, and then did a PhD in the UK in computational neuroscience. I then did a postdoc also in the UK, and another one in here in New York with Bill. And I then moved to a faculty position, and I started my lab a year ago as an assistant professor at SUNY Downstate. So my interest is mostly on this idea of large-scale biophysically detailed models of motor cortex, now moving on to auditory cortex as well, thalamus, and as well, of course, using them to understand neural coding, disease, and potential treatments for disease, as well as developing these tools to build this kind of models. Any volunteers? Bill?
I can go next.
I'll introduce myself. Or Joe, you want to go first?
No, go ahead, Bill.
Okay, so Bill Litton, I've been teaching courses, mostly NEURON, because NetPyNE is relatively new, for this past 25, 30 years. And I used to say that since I wasn't the oldest person here, I would really benefit from the advice and suggestions of the older people. But now it turns out really what I could benefit is advice and suggestions from the younger people. So that's changed slightly, but it's still, I would encourage, at least for my talks, I think other people's talks, to really try to interrupt during the talk, especially for me, at least in this, Zoom has now become a standard verb. So I'll just say Zoom type meeting. It's just so much nicer to know that somebody's paying attention. At least, you know, when you have an audience out there, you can look at people and count how many are asleep, which is a good indication. Anyway, I've been doing this, as I say, for many decades. I'm a neurologist by training, have an MD, did a neurology residency. I still see patients, but really 90% of my time is spent doing research. I've been doing research primarily, I'd say, over these many years on single cell modeling, but more and more on network modeling for the past 10 to 15 years. We got a big boost through a DARPA grant that we had, and that's what enabled us to hire Salvador and a bunch of other people who have been really fantastic in moving this forward. In terms of wrappers onto NEURON, really, NetPyNE is in a sense a wrapper on top of NEURON, which remains the simulator. We've been thinking about this, I really remember quite clearly, since 1988, working with a guy named Jack Wathy, who I think eventually went into ligand receptor interaction modeling. And he and I, mostly him, frankly, he was a great programmer, put together something in SCCS, which was the first of the version control systems, and that would, enable you to package and run a simulation, really just single cells at that time. So this has been something that's finally achieved fruition thanks to Salvador decades later. I focus, as I say, well, I didn't say, is on neurological and psychiatric diseases, brain diseases, since I am a practicing doctor, but of course also fascinated by what and how the nervous system works. And that's, I think, enough. I'll let Joe have it.
Hello. So I'm Joe Graham. I'm from Arizona. I studied neuroscience at Arizona State and bioengineering. I worked in a spinal cord injury lab where we worked with a rat model of spinal cord injury, and we're interested in locomotion and interventions that can improve locomotion after a spinal cord injury. So we did a lot of EMG and biomechanics stuff, as well as the neuroscience, recording from spinal motor neurons in vivo. And then with all that data, we wanted to create computational models, neuromusculoskeletal models, which is really my passion. And then in that process, we needed actual morphological motor neurons because spinal cord injury changes their morphology. We didn't have any available, so I started developing algorithms to grow them. And that worked pretty well and landed me a job at the Blue Brain Project, where I started growing cortical neurons. And then I've always been interested in multi-scale, large-scale modeling, especially towards neuromusculoskeletal. So I got a job with Bill Litton at the NeuroSim lab, where I used NetPyNE to start developing some cortical network models to explore different topics. I enjoyed NetPyNE so much that I applied to work with Salvador and become a NetPyNE developer. And so I've been in Brooklyn, New York for the last year working as a NetPyNE developer. And I look forward to working with all of you.
Salvador, a question. If you stop sharing, maybe I can see more faces. I can only see a few faces now. But again, small screen.
Hello, everyone. My name is Fernando. I'm from Brazil. I'm a physicist. I did my PhD in physics, and I did another postdoc in physics, studying mainly integrated fire neural networks, studying synchronization and synaptic plasticity. After the first postdoc, I did another postdoc in the lab in the electrophysiological data analysis, and they work in many kinds of the data analysis using multi-electrode array, intracellular array, many kinds of the analysis in the lab. And since last year, I'm working in the Salvador lab, studying mainly multi-compartmental models and the networks. My first focus is in somatomotor cortex, and using NetPyNE to reproduce the somatomotor cortex.
All
All right. Hello, everyone. My name is João. I'm also originally from Brazil. I'm doing a PhD in SUNY Downstate, working with Dr. Salvador Durber now. I have a bachelor's in biomedical engineering, but I kind of like always wanted to pursue something into neuroscience. So I did a master's in neuroscience in Brazil, but like working with biofeedback devices, developing more of like a human-machine interface. And now in the PhD, I finally made the transition to actually work with computational models, which is something I got in contact with during LASCON, which is a summer school that we have for computational neuroscience in Brazil. So I met Bill, Salvador, and Erica there. It was a great experience. And then I kind of jumped into this field, had a chance to come here to do a PhD in New York. So yeah, I'm one of the tutors and happy to meet all of you guys.
Hello, everybody. My name is Erica Griffith, so I am one of the tutors as well. I'm an MD PhD student at SUNY Downstate, and I'm currently doing my PhD with Dr. Litton, with Bill, and with Sam. I'm not sure if he's here, but Dr. Nemoten, who I believe will be part of the course as well. And I've been working closely with Salvador, who's great. You guys are all in for a treat. He's a really great teacher, and we have a really great crew here. So I've been working on a multi-scale circuit project on the auditory cortex with Salvador that I'll also be talking about later in the course, and really look forward to working with all of you. Nice to meet you.
Hello, I guess I'll pop in here. Everybody see me? Yes. Hi, I'm Donald Doherty. I got my PhD in neuroscience at UC Irvine way back when, and did a lot of recording out of actually Barrel and other areas of the brain, pretty much every area actually. But anyway, I actually left after a postdoc with Dan Simons in Pittsburgh. I left and started a company and was an entrepreneur for many years, and then came back actually just a few years ago and ended up with this great group here. I've been working on with the motor cortex model that Salvador showed, and doing avalanches. And so it's kind of a very interesting sort of high level theoretical approach onto it. So anyway, I'll be very excited to work with all of you.
So hi, everybody. I'm Robert McDougall. I'm an assistant professor in the Health Informatics Division of Biostatistics at Yale. So I actually started out as a math major, CS minor in undergrad, was math all the way through PhD, although I was looking at, my advisor was Dave Terman, who was the, I think, senior associate director at the time of the Mathematical Biosciences Institute at OSU. And so he was very interested in neuroscience questions, remains very interested in that. And so we looked at, amongst other things, calcium dynamics, since that was brought up. And one of the things that we found when I was trying to do some of these questions was there really wasn't a great tool out there to answer some of the kinds of questions that we wanted to study about how calcium waves travel, for example. And then so I went to the Computational Neuroscience Conference. It was my second conference I ever went to. I went on Facebook. I found a random roommate who was this guy, Sam Nemoten, who's mentioned already. And so Sam tells me that his advisor, Bill, is very much interested in these sorts of questions. And I said, and that he had even organized a workshop at the conference, and that I should go to that workshop. And I did, and I started talking to Bill, and through that, I got involved in a project with Bill and Michael Hines, who's the original developer of NEURON. And so an interesting set of series of things happens after that. CS postdoc, neuroscience postdoc, medical informatics postdoc. Got another master's in computational bio while being a postdoc. Some time in neurosciences research faculty before moving over. But for the last 10 years now, I've been a developer for the NEURON simulator. And for most of that time, I've been a developer for ModelDB, which is a discovery tool for computational neuroscience models. It's been mentioned. Certainly happy to talk to anybody about anything with NEURON, ModelDB, or informatics. Love talking about informatics, which is my other area of research interest. But I'll leave it at that for now, and you'll see more of me later today.
Hi, everybody. I'm Craig Kelly. I'm going to be a tutor for the course. I'm originally from Massachusetts. Did my undergrad at Boston University. Took a couple jobs where I was sort of the computer guy in some neurophysiology labs. Did some projects on like Alzheimer's disease, recorded some local field potentials. Then went to a place that did more like learning and memory stuff, got thoroughly indoctrinated in the sort of cult of the hippocampus. And now I'm a PhD student here at SUNY Downstate in Bill Litton's lab, working on two sort of disparate projects. One's about biophysics and inductance and impedance and neocortical layer 5 pyramidal cell dendrites, and the other one's about cortical spreading depression. And that one involves a lot of the reaction diffusion stuff and working with Robert and Bill and some other folks on that. Adam, who I don't think is here. So yeah, looking forward to working with y'all, and yeah.
And Sam, I think we have.
Hi, so I'm Sam Nemotin. I was a while ago. I got my PhD at the Neurosim lab with Bill Litton, and I was doing a postdoc. At that time, I was doing computational modeling of neocortical circuits and hippocampal circuits. And eventually, we were working on RxD, also like Robert mentioned, so the calcium waves and other neocortical circuits, looking at different intracellular signaling pathways. And more recently, I'm at the Nathan Klein Institute working with Sal and Erica and Bill and Peter Lakatosh there on a detailed model of the auditory thalamocortical system. So we're integrating some experiments with non-human primates, collecting data from their auditory cortex and thalamus, and then using that to constrain the model and trying to understand mechanisms for auditory processing, including speech processing. And I'm also working on another project to model reinforcement learning and to teach a model of the visual and motor cortex to play games, basically. So that's with Haroon, and he's going to give a talk on that later in the course as well. So that's basically what we're working on now. And look forward to hearing more in this course and the different presentations.
And Sam lives in a house full of smoke.
Yeah, or underwater, actually.
I thought he was in the witness protection program.
Don't tell anyone. Yeah, too late.
Thank you, Sam. So yeah, thank you, everyone. I think we can stop for now. We have, in theory, the next lesson starting in 5 minutes. So maybe we can start actually at 11:35 to give a bit of a break. And we'll see you all in a bit. Thank you so much.